Hello everyone, I'm Priyanka and today we will talk about equipment used in clinical microbiology laboratories part 2. So, next we have a magnetic stirrer. Magnetic stirrer is a device commonly used in microbiology laboratories for the purpose of mixing liquids. Talking about the working principle, this device consists of a rotating magnetic or an electromagnetic creation a rotating magnetic field that allows the stir bar, a piece of heavy metal, to move around in the vessel. It is coupled with a heating system to heat the liquid while it mixes. Talking about the uses, it is usually used for mixing various liquid components in the mixture in a chemical or microbiological laboratory. This device is used in place of other stirrers as it is noise free and because the size of the stir bar is too tiny, there is less chances of contamination. So this is a magnetic stirrer. Next we have microscope. Microscope is a device that allows the observer to an excreting close view of minute particles. Talking about the working principle, there are many different types of microscopes, each of which work on their respective principle. However, there is some commonality in them. The basic principle in a microscope is magnification. Based on the relative position of an object from the lens or electromagnetic magnets, different position, nature and magnification of the image can be achieved. Different type of microscopes are developed to cater to the specific needs of observation. However, the common theme is magnification. Talking about the use based on the type of the microscope, different microscopes are used for different purposes. They are primarily used for the observation of minute particles which cannot be observed with naked eye. So this is a microscope. Next we have a pH meter. pH meter is a device used in laboratory that measures the hydrogen ion concentration in water-based solution to determine the acidity or alkalinity of the solution. A pH meter is often termed as potentiometry pH meter, as it measures the difference in the electric potential between the reference and the pH electrode. Talking about the working principle, in a potentiometric pH meter, a single or multi-glass electrode connected to the bulb selective to hydrogen ion are attached to the metal rod. When the bulb with the electrode is dipped into a solution, hydrogen ion in the solution exchange with positive charge on the electrode, generating an electrochemical potential which is displayed in terms of pH unit on display. Talking about uses, a pH meter is primarily used to measure the acidity of the pharmaceutical chemical, culture, soil and water treatment plants. It can be used to measure the acidity level in wine and cheese during the production. So this is the pH meter. So next we have spectrophotometer. The spectrophotometer is an optical instrument for measuring the intensity of light in relation to the wavelength. Based on the amount of the light absorbed by a color solution, a quantitative analysis of solution can be done. Talking about the working principle, spectrophotometer is based on Beard's-Lambert law, which states that the absorbance of light of a solution of a particular wavelength is directly proportional to the concentration of the substance and the path length. Different wavelengths of light are passed through a solution as different substances have better absorbance at different wavelengths. Based on the absorbance of the particular wavelength, the quantitative analysis of the solution can be done. Talking about the uses in the microbiology laboratories, a spectrophotometer is applied for the measurement of the substance concentration of protein, nucleic acid, bacterial growth, and enzymatic reactions. So this is a spectrophotometer. Next we have Vortex Mixture. A vortex mixture is one of the basic technologies used for the mixing of the sample in the glass tube or a flask in laboratory. Talking about the working principle, it is based on the simple principle of causing reactions and homogeneations by agitating the mixture. Motorized draft shaft present on the mixture oscillates and transfers the movement of the sample tube, causing the sample fluid to undergo turbulent flow. Talking about the uses, vortex mixture is mostly used for mixing of the various sample fluid in the sample tube and also allow for the homogenization of the cell and the cell organelles. So this is a vortex mixture. So next we have water bath. Water bath is a conventional device that is used for chemical reaction that requires a controlled environment at a constant temperature. Talking about the working principle, a sensor in the device transfers water temperature to the reference value, which is then amplified and control system generate a signal for the heating system, which heats the water to the desired temperature. Talking about the uses, water baths are primarily used for heating samples under a controlled temperature. These are suitable for heating chemicals that might be flammable under direct ignition. So this is what talking about the water distillator. A water distillator is a device that purifies water by the process of distillation. This instrument is commonly used in the medical laboratory, microbiology laboratories, organic chemistry laboratories, and medical industries. Talking about the working principle, a water distillator is based on the principle of distillation. According to this process, water is first brought to the boiling and then condensed into a liquid form to obtain a pure distilled water. Talking about the uses, it is used to obtain distilled water required for many lab tests as well as for the preparation of culture media. So this is the distillation unit.